So we are very delighted to have our second speaker, Professor Claire Adjiman from Imperial College London. Claire obtained her Master of Engineering from Imperial College and then a PhD from Princeton. He worked with the famous late Professor Fludas at the time. And since then, she has done some really fantastic work on using basically applied math and optimization algorithm to study different types of systems. She has won multiple awards and have a lot of memberships in professional societies. Some of the awards that she has won include RA Engineering ICI Fellowship, the Philip Leverhulme Prize for Engineering, the SCI Armstrong Lecture, the EPSRC Leadership Fellowship, and the Royal Society of Chemistry Elizabeth Colburn Memorial Lecture, and the American Institute of Chemical Engineering Computing and Chemical Engineering Award. So without further ado, I'm gonna leave the floor to Claire to talk to us about a hierarchical approach to crystal structure prediction. Or actually my, what happens? There is this thing that's just limiting my prediction, status, applications, and challenges. So Claire, the floor is yours.
Thank you, Amir. It's a very long title. Thanks for the really kind introduction, the invitation, and also thank you, Liz, for your very interesting talk. I think it makes more sense to ask questions after I've presented my talk so that I can give a bit of an introduction to what we're trying to do. So it's going to be a little bit of a whirlwind tour of what we try to do in crystal structure prediction. So you maybe will be familiar with crystal structure prediction for proteins, protein folding, but we're working here with smaller molecules, typically organic molecules, motivated in particular by the pharmaceutical industry, where the active pharmaceutical ingredients, APIs, are usually in crystal form, which means you can take a tablet, which is much nicer than an injection or something like this. But the problem with solid phases is that the properties of your drug are really defined not only by the molecule itself, so you have here a small molecule called ROI, but also by the specific crystal shape that it takes. So there's a bit of a connection here in terms of structure and shape with Liz's talk. So ROI is particularly famous because the molecules can pack in many different ways. So each periodic packing of these molecules represents a different crystal structure. ROI has many. In fact, new ones are being discovered. I hear there's a new one coming out soon. But this in 2000, those were the ones that were known at the time. And you can see that their aspect ratio, their color is quite different. And from a pharmaceutical perspective, that also means that those crystals are going to dissolve at a different rate or are going to have different level of solubility in the gut, and that impacts how much drug gets through into your bloodstream and therefore how effective it is. So understanding what crystal structures can form is really important in the development of a solid product like a drug, but also agrochemicals, all sorts of other applications. It's really important to therefore be able to identify all of the forms that might occur in manufacturing or indeed during storage of the product. It's quite challenging to do so experimentally because thermodynamically, although the most stable form at given temperature and pressure will eventually appear, it can take a very long time to do so. So you might be making a certain form for a couple of years, and then all of a sudden a new form appears. And that has happened in the past and cost hundreds of millions of dollars in interruption and trying to fix this issue, but also, of course, been really impactful on the patients who did not have access to their drugs anymore once a more stable form was formed. So we want to be able to help the understanding through computational methods, and we also want to be able to support the development of drug products by using the predictive tools to make decisions. So just to define the problem, so we're looking at a molecule, so represented here, that is going to adopt a certain shape itself is a conformation in the crystal in terms of how the various parts of the molecule are arranged together. So sometimes you have molecules that are very rigid and won't change in the crystal, and sometimes they're a bit flexible and they can adapt to allow different packings. So we're interested in the position of all the atoms in the molecule, the relative orientation of the molecules in the repeating unit, the unit cell. And then we're interested in the size and shape of this repeating unit. So that would be defined by some unit cell lengths and some angles as well. And what those molecules will tend to do is to find a stable state, which in terms of fixed temperature and pressure and composition, if you have multiple species in the system, means that you're trying to minimize the Gibbs free energy of the system. And in order to make the problem tractable, we're going to make some simplifications because it's very difficult to compute the Gibbs free energy. So I'm just going to go through this quite quickly. We're going to neglect essentially volume effects. We're going to neglect temperature and entropic effects here because they tend to be quite small. And we're going to assume we're in fact working at 0 Kelvin. And we're going to focus on the lattice energy rather than the minimization of the Gibbs free energy. And we are going to split in the method that we have been using. We're going to split that lattice energy into different components. One that is the intramolecular energy, which actually refers to the molecule on its own and the conformation that it adopts and how much that deviates from the conformation it would have in the gas phase in complete isolation. And that in gas phase, it's at most relaxed, lowest energy. And so there's going to be a penalty to pay for this deformation, and that's delta U intra. And then we have two other components that we're going to take into account. An electrostatic component that is to do with the electrostatic field around each molecule and how they interact together because of that. And these are quite short range interactions typically, and then much longer range interactions. In this term, well, there's longer range dispersion interactions, and then also the repulsion that occurs when two molecules are very close together. So we're looking at these kind of two ends with this additional term here. Okay, so the model that we use here in crystal structure prediction, the lattice energy model, is really central to developing a method that is going to work. Typically, as we search for potential crystal structure, we are going to generate hundreds or thousands of plausible structure that are low-lying minima on the lattice energy landscape. And we do want to find more than one. We're not just interested in the global minimum because due to kinetic limitations, the molecules might go to the form that is not the most stable at given conditions. Okay, so it's quite important to get them all. And we want to generate, therefore, a landscape, so essentially a list of structures with different energies. And we need to rank them correctly in terms of the energies. And so we need something that is quite accurate. So we need efficiency because of the scale of the problem we're looking at, but also accuracy, enough accuracy to achieve this accurate ranking at the end. Unfortunately, because the atoms are quite close together, we can't get away with too many approximations here. So to get a really accurate ranking, sufficiently accurate ranking, I should say, for applications, we really need to use quantum mechanical methods that take into account the whole of a periodic structure to, essentially to infinity. So these, as you might imagine, are quite expensive to compute. And so what we're going to try and do is to develop some initial models that are like force field models that are a bit less expensive and enable us to screen through potential structures to narrow in on the most likely ones and do the expensive calculations only on those. Okay, so if you compare the two approaches, force fields versus the DFTD calculations, so you have a typical form of a force field here, as the one I've shown you on the previous slide, and then where theta denotes the isolated molecules, so the conformation of the molecule, and X denotes essentially the shape and size of the unit cell. And so in the case of the DFTD calculations, this is really all mixed up together. We have the opportunity to do a bit of separation when we're looking at force fields. Unfortunately, with DFTD, we're talking about thousands to tens of thousands of CPU hours per structure. And But the good thing is that we get reliability, and we have the opposite behavior with force fields. So it can be extremely cheap, but it's not very reliable. Okay, so we're always trying to achieve a balance between those two. So the method that we've developed to try and achieve that balance is a multi-step hierarchical approach, as the title of my talk indicated. And I'm going to try and walk you through this. So we start with a 2D vision of the molecule. So we don't know anything about conformation, just how the atoms are connected together. And this is the Roy molecule that I've shown here. We are going to do some quantum mechanical calculation on the isolated molecule. These are much, much cheaper than periodic crystal quantum mechanical calculations. And we're going to look at how the energy of a molecule, the intramolecular energy, varies with conformation. And we're going to obtain that energy, but also how the charge density, electrostatic potential varies around that molecule in isolation. We're going to use this molecule-specific information to develop a tailored intramolecular and electrostatic potential. We're going to combine this with a transferable potential for the repulsive dispersive term that I've mentioned. And that is going to make up our lattice energy model, which we're going to use to launch a global search. So we have a code for this that really tries to find as many structures as possible. We typically generate 500,000 to 2 million structures with this code. So really quite a number of calculations. We'll then take the lowest 1,000 or so structures, up to 7,000, I think is the highest number we've been to, and take this to a second stage where we take the same model as before, but we improve it. So for example, we take into account more flexibility of the molecule than we had previously. We improve the electrostatic potential. We go from point charges to multiple description of the electrostatics. And through this, we get much more accurate, more expansive as well, lattice energy calculations, and we rank those energy structures. And then we can take this to the next stage, and here we have a number of options that enable us to get a much more accurate ranking, including the DFTD models that I've mentioned, but there's other things that we can do. And then we get a list of structures that hopefully contain the polymorphs of this of the compound of interest. So that's the overall structure. And what I want to do now is to go through these stages in a bit more detail to try and explain what we do at quite a high level, and also highlight some of the challenges that perhaps we can discuss later. So I'm going to start by focusing on the tailored component of the potential, so the intramolecular and electrostatic potentials. So our lattice energy model is the one shown here, and I'm focusing on these two terms here. So I'm going to be interested in minimizing the lattice energy with respect to, so in the overall model, the lattice energy with respect to the conformational degrees of freedom, so conformation of the molecule. So I'm going to make that my kind of main variable in the problem, trying to reduce the size of the problem a little bit. The intramolecular energy depends on those variables only, and the intermolecular energy does depend on those, but also on the lattice variables. And so I'm going to do an inner minimization with respect to the lattice variables for fixed conformational variables. And I'll explain some of the other components of this term here. So the problem here is that the intramolecular energy as a function of the conformations, and so one of these other terms, which is the electrostatic potential as a function of these conformational degrees of freedom, are very expensive, even for an isolated molecule, much less than the FTD, but they still can require a few hours of CPU time. And then in terms of the scale of the problem, what we can do here, if we do quantum mechanical calculations for each value that we want to consider, is very small in terms of the scale of the molecules and the number of flexible degrees of freedom. So we need something a bit better than many quantum mechanical calculations. And so what we've developed is a concept of local approximate models. So these are kind of surrogate models. And I just want to describe how we've developed them and a little bit why we have chosen this. So they're going to give us the full space of conformational degrees of freedom, the full description of the conformation of the molecule, the delta U intraterm, and the electrostatic potential as a function of these selected degrees of freedom. So we can derive these models, and I will show you their form in a minute, either a priori, so we can build up a database of these models, for example, on the uniform grid, or we can do them on the fly. So as we sample a part of space that we hadn't sampled before, we can generate a new QM calculation as and when required. We can choose what we include in this set of degrees of freedom. And what we do in practice is that we always reuse our calculations. So anytime we do a QM calculation, we store it in a database and we then reuse it later. And we can reuse it often in the global search, but also in the refinement stages, at least the first refinement stage. Okay, so this reusability is really important for efficiency. So let me say a little bit more about these variables, the conformational degrees of freedom. So we identify initially a set of independent degrees of freedom, which are only torsion angles at the global search stage, and then later can be bond angles if we want to be a bit more accurate. And they have a strong relationship to the intramolecular energy. So changing these torsion angles really has a big impact on the value of the intramolecular energy. And then all other variables that describe the conformation of a molecule are what we call dependent degrees of freedom, theta r, for the conformation. And then the electrostatic potential is also dependent upon these here. So we essentially develop a reduced space model for this. Okay, so I wanted to give you a little bit more detail so that you understand the computational cost. So every time we take reference points that are going to be the points where we have our local approximate model, and at each value of the reference point, we do a minimization of the intramolecular energy with respect to the dependent degrees of freedom. This gives us the energy at that point. It also gives us the solutions, so the theta r's and the electrostatic potential. Then as we consider a point, so this will be the red points here, we consider a point that is not too far from that, we can evaluate the energy and the dependent degrees of freedom right here. So at that point, theta f, by essentially doing a second order Taylor expansion, taking into account the optimality conditions that we had obtained here. And so we have here some vectors and matrices that are obtained at the solution of this point. Okay, and so it's just a simple expression, quadratic expression in this case. And then for the degrees of freedom, the other geometric variables, we have a first order expansion. And actually for electrostatics, we just assume that they don't change very much as we move away from this point here. Okay, so this gives us a nice recipe to calculate all of these quantities as a function of our very small set of conformational variables. So we initially did this on a uniform grid, but this can become quite expensive quite quickly as you increase the number of degrees of freedom. So we've done a number of things over the last few years to try and accelerate this. We've used adaptive generation of the points. So we start from the uniform grid, but you can see here, so with these two torsions and the energy. So the lower the energy, the better. And so we are sampling mostly in the space of low energies, and that can help us to achieve a 70% reduction in CPU time for highly flexible molecules. And you also find, because we're using, these are very localized models, so at the boundaries between the models, when you move from one to the other, you find that you have some discontinuities, which of course creates a lot of problems when we're doing local minimizations. So we've introduced an exponential function to actually take into account multiple local models and combine them together. And you can see that from something that was a lot of discontinuities on the left, again, two torsion angles here, and the energy color coded, you get something that is much smoother. And again, that buys you a very significant reduction in CPU costs, 65% reduction. And then finally, we've done some partitioning here. So where you have a rigid core somewhere in your molecule, you can treat the flexible torsions as almost independent of one another. And so instead of sampling in the two-dimensional space here, we sample in two one-dimensional spaces here. So of course, for such a small example, it doesn't have, it doesn't make a big difference, but it does have a big impact once the number of degrees of freedom starts to grow. One of the challenges we're facing, and which I want to put to the audience, is the cost of the lamp generation. So as you saw, we have a second order Taylor expansion. So we need some Hessian matrices for that. So we're calculating those Hessian matrices at the level of quantum mechanics. They're quite expensive. So here, taking a small molecule, methylparaben, if I just look at the lamp database generation, it doesn't really matter if I've got one torsion group or if I just partition it, about 100 to 100 or so CPU hours. And the global search is about 10 times that. So like a much bigger molecule, this is molecule 20 from one of the blind tests on crystal structure prediction. You can see that the lamp database generation really is what dominates the search. Of course, here, so these are my global search and refinement stage. I can search longer if I want to. I could spend a lot more time here, but in any case, this really dominates the cost, and that becomes a big issue as we try to deal with bigger, more flexible molecules. So that's one challenge of these models. Then looking to the repulsion dispersion potential. So these are transferable models. And so what we've been doing in recent years is to develop a way to estimate those models in a better way to increase the accuracy. And this can be used at the global stage, at the refinement stage, and at the final refinement stage, in which we might actually tailor the model to the data that we've generated, the crystal structure we've generated for a particular molecule to improve its accuracy. Okay, so we're actually using computed crystal structure data at the DFTD level to parameterize these models and improve their accuracy. And so here we have some nice, very large-scale optimization problems. So we have a training set of reference crystal structures. We have a form of our potential, and then we run a parameter estimation where we're trying to match the geometry of the crystal structures and the energy of those crystal structures. And so we do that mostly with rigid molecules. So to evaluate this goodness of fit or the least squares function, rather we have to run a minimization of the lattice energy. So many minimizations within our parameter estimation, and we get our optimal set of parameters. Okay, so here we've been able to develop this. We're able to do this for quite large systems. I need to move my Zoom window here. But we have, for example, over 300 training structures, quite a large number of parameters. And really what we're looking at now is to understand better the statistics of these models so that we can train them better. Okay, so we've done quite well in terms of the non-hydrogen bonding structure. So this is the energy of the reference structures versus the calculated energies. And you can see here we're doing quite well. But in terms of the hydrogen bonding structures, we're not doing so well. And it's something that we're working on in the group now to try and improve. And I think what might be interesting here is to maybe try and analyze structures sort of on the fly, maybe with some of the tools that Liz has been talking about. I don't know if it's possible to adapt them to this, to try and understand the connections between different structure and to be able perhaps to adapt the model to the structure. Then thinking about the next stages, so we've got a model, now we need to use it to do a global search. So I'm looking at time. Until when do I have, Amir?
Roughly 3 minutes, I would say.
Okay, great. So the CrystalPredictor algorithm enables the global search. And so one of the big aspects here is that we generate structures using a Sobol sequence in the space of potential unit cell parameters, positions and rotation of the molecules and the torsion angles, and also in the space of space groups that we might have. So one of the issues that we've been facing here recently is as we try to look at unit cells that have more molecules in them, so what's called Z'2 or greater, we run into issues to do with molecules overlapping as we place it and we try to generate structures. And so without looking at all the numbers here, really the key challenge, which I'll be happy to discuss, is how do we generate conformations and crystal structures to start our local optimizations that do not overlap and avoid wasting a lot of time here. I'm going to skip this on the local search, but I just want to use this slide to emphasize the importance of efficiency through the molecule, and again go back to this issue of the lab models. Okay, so we are generating more and more refined landscapes, energy landscapes for our structure. So we don't want to miss anything. So at the beginning, we need to be broad enough and sufficiently accurate to make sure that the low energy structures are going to be the experimental ones. But yet we want to re-rank and re-rank. So this is a relatively small molecule, molecule 31. And I just indicate the different stages here, how many structures we've minimized. And so you can see we do a lot to start with, and then that reduces significantly. And then essentially the cost per structure. So VASP is the DFTD method. And so you can see that really grows exponentially. And we actually have, quite big gaps at each stage of a methodology. And I think any method that we can use to improve the reliability of the methods at each stage, or perhaps some intermediate methods to try and achieve better ranking, better correlation between the rankings at each stage would be useful. But you can see it's quite infeasible to do many structures at the final stage. This is just 46 structures that we've been able to do in this case. I think I'm going to skip this. This was really a call to say an example of an application to say that speed is really important, and this idea that we're working with isolated molecules enables us to go towards the design of crystals. So to identify potential co-crystals that you might be able to observe experimentally, which are the ones in purple here. They have lower energy by our model, whereas others here have higher energy and are not observed experimentally. And so by applying these tools, if they're fast enough, you can start to design combinations of molecules to achieve a certain effect. So I just want to summarize by saying that we have had quite a bit of evolution of CSP methods over the last few years, trying to balance accuracy and efficiency. I think there's a lot more we can do to do this for molecules of practical importance, especially as the pharma industry moves towards things like peptides and more complex molecules, especially around accelerating surrogate model construction and improving the generation of structures. That I'd like to thank you for your attention. I'd be happy to answer questions, and I'm really looking forward to the discussion.
Thanks, Claire, for the fantastic talk. So there were a lot of stuff in the chat, so I might have missed any questions that people might have written in the chat, but yeah, please read. Yeah, Sapna, are you up? Okay, so sorry. So let's give Claire a round of applause and raise your hands if you have any questions. So I just have one question myself before we get other questions from the audience. So as I understand, the unit cells that you're considering are mostly like one molecule per unit cell, right? And we know that we can have like more complicated unit cells. And I wonder whether you have considered them, how much more difficult does it make the whole problem, and whether they are even relevant for in pharmaceutical industry.
Mm-hmm. Okay, so I'll start with the last question. Yes, they're absolutely relevant for the pharmaceutical industry. I think the most we've gone is to 4 unit cells, so there's no theoretical limit to the number of molecules in the unit cell in our methodology, in the asymmetric unit cell, in the method that we have. The challenge is in that you have a much bigger space of possible crystal structures. So you need even more efficiency there. If you consider a Z'4 space, actually that includes all the Z'3 structures and all the Z'2 and all the Z'1 in there. So it's a very big space. I don't think we search it very efficiently currently. And then we run into this issue, especially if you have large flexible molecules, that a lot of the structures we generate have overlaps. And so we're wasting a lot of time generating structures that go nowhere. So there's a lot of room for improvement there.
I mean, and to that, I think Elise, do you have a question, or Aurora? I didn't see who raised his hand.
I have a question.
Yeah, sorry, go ahead.
This was a lovely talk, and it's an area that I'm really interested in. I'm curious though, because and maybe I missed it, but you never account for solvent effects. And so I don't have a good understanding of the role to which solvent influences the relative energetics of these, or incorporated into the crystal structures and things like that.
Yeah. Yeah, so that's a great question. There are a couple of aspects there. So we're looking at the solid state. So you can have a solvent in your structure, that'd be a solvate, and so so you would have at least 2 molecules in the asymmetric unit, right? And so we can, just like I flashed some coformer a coformer slide. You can have 2 molecules, and we can predict solvates. You have to sort of set out to do that, so you have to go, Z prime 1, I'm going to look for all of them, Z prime 2, and then you have to look at different stoichiometry. So you could have, 1 to 1 or 1 to 2 or 1 to 3. So each of that would be a separate search. We haven't looked at how you would make that more efficient than just working through all of that. You have even in the Z prime 1 landscape that we generate, you will have some fairly high energy structures that are dissociated structures. So there will be structures where there's only the drug molecule, let's say, but there's quite a few gaps there. So that would be where a solvent might have been. So that's possible. Then in practice, the solvents will influence which crystal structures you see in reality. All right, so they don't influence the intrinsic stability of your crystal structure, but you might see different crystal structures crystallize out of solution depending on the solvent that you have, and then eventually one structure, if it's a metastable structure, eventually convert to the more stable structure, but that could take years. Yeah. And we have not looked at all at ways to predict. I mean, the whole field of crystal structure prediction, nobody yet can tell how do you actually make the crystal structures that we predict exist. That's a big challenge.
Thank you.